Hello guys everyone. I welcome you all to Unacademy. Good evening everyone. अगर आप लोग मेरी आवाज़ सुन पा रहे हैं तो कमेंट बॉक्स में हाय टाइप करिए, हेलो टाइप करिए, मेरे को पता चल पाए कि आप लोग ऑनलाइन आ चुके हैं. Everyone. So guys, let me begin today's session. You all are welcome. You need to go to Google Play Store where you will be finding an Academy learning app. You need to install the app and you need to select your goal. In the select your goal, you need to select CSIR UGC NET. So once you select that category, you will be getting access to various courses from many top educators for the CSIR UGC NET category. And if you want to get complete course, you have to get the CSIR UGC NET subscription. Now the subscription is for 1 month, 3 months, 6 months, 12 months and 24 months. I would suggest that you get a subscription for 12 months to 24 months because the classes will be going year long and you will get enough time to understand what is happening in the course. You can interact with the educator, you can practice so many live mock tests, questions, quizzes and so on. So it will be very helpful if you get the 12-month subscription, uh, and you can always use my referral code HY10. So you'll be getting a discount of 10% in that. So you can confirm the details. You can go for the CSIR UGC NET subscription for 12 months. You can proceed for the payment, and once you proceed to the payment, you will be getting a discount of 10%. So the entire course you can get for 10,395, and you can do the payment either by card or debit card, credit card or debit card, or by EMI, and these EMI are no cost EMI. You can also pay it by net banking. So this is all, and I would like to mention that we'll be holding classes live on YouTube as well, like this session. So both are free classes as well as paid classes. You can take the access and improve your learning. So let's begin today's session. Today's session is about enzymes. So as you all know that enzymes are biomolecules which are proteins in nature, and they have a high affinity for a particular substrate, and the enzyme acts on a substrate to form the product. So let's study. And enzymes are important, they are key to life. No enzyme, no life. So this is an important statement, because most of the proteins which are present, most of the enzyme which are present in the body are made up of proteins, and if enzymes are not working, various uh, life processes, various life cycles are not taking place properly, and it becomes a problem. So no enzyme, no life. It is a true statement. So no and no. So enzymes are key to the life, and no enzyme, no life. This is a true statement. So let's begin. So what is an enzyme? Enzymes are the globular protein which function as biological catalyst. They speed up the reaction rate. They speed up the reaction rate by lowering the activation energy without being affected by the reaction it catalyzes. So this is an enzyme, as you can see over here. This one is the active site. This one is the active site. This is the active site where the substrate will come and act. So they are basically globular proteins which functions as biological catalysts. They speed up the reaction rate by lowering the activation energy without being affected by the reaction it catalyzes. Enzymes are protein in nature. They are globular protein. There are also ribozymes that are RNA molecule with enzymatic activity. They undergo catalytic behavior of an enzyme depending on its primary, secondary, tertiary, or quaternary structure. Enzymes of digestive tracts and those found in blood are present in inactive form called zymogens or proenzymes. Now, what is the active site? So, the active site, each enzyme has an active site where the substrate binds. Active site is the region on the surface of the enzyme to which the substrate will bind and catalyze the chemical reactions. Enzymes are composed of long chain amino acids that have folded into very specific three-dimensional shape which contains an active site. So this is a substrate. This is a substrate. If you can see, this one is the substrate, and this is the active site, and this is the enzyme. Enzymes are highly specific for the type of reaction they catalyze and for their substrate. So there is a high degree of specificity for the reactions they catalyze and for their substrate. Enzymes are highly specific in nature. Enzymes with highest specificity and accuracy are involved in copying and expression of the genome. These enzymes have proofreading functions such as the DNA polymerase. Now, enzymes are also found involved in the copying and expression of the genome, such as the DNA polymerase or the RNA polymerase. So these enzymes have a high turnover number. They have a high specificity and accuracy. So some enzymes are require non-protein molecule for their activity. So enzymes are conjugated protein known as holoenzyme. So some of the enzyme they require non-protein molecule for their activity. So enzymes are conjugated protein known as holoenzyme. Now holoenzyme is formed by apoenzyme plus the cofactor or the coenzyme. So enzyme without the non-protein part is inactive and it's called the apoenzyme. Cofactors can either be inorganic or organic compounds. Cofactors can either be inorganic or organic compounds. Enzyme without the non-protein part is inactive and called as apoenzyme. So holoenzyme is apoenzyme plus the cofactor or the coenzyme part. Good evening, Shatruhan Patel. Good evening. If you are able to hear me, then please type hello. Everyone who is there online, please type hello. so that I can get to know that all those who are attending the session, if you can hear my voice. अगर आप लोग हमको सुन पा रहे हैं, मेरी आवाज़ को सुन पा रहे हैं, तो आप लोग हेलो टाइप करिए, जिससे कि मेरे को पता चले कि आप लोग ऑनलाइन हैं इस समय. Good evening, everyone. So enzymes require non-protein molecule for their activity. So enzymes are conjugated protein known as holoenzyme. Enzymes without non-protein part is inactive and called apoenzyme. Cofactors can either be inorganic or organic compounds. Mechanism of enzyme action. The enzyme reaction takes place by binding of the substrate with the active site of the enzyme molecule by several weak bonds. So enzyme plus substrate will get the formation of enzyme substrate complex, and enzyme and proteins are enzyme plus product is formed after the reaction takes place. So there is a particular mechanism of enzyme action. There is a particular mechanism of enzyme action. The enzymatic reaction takes place by binding of the substrate with the active site of the enzyme molecule by several weak bonds. So Dashrath Hansa, hello. Marina Boro, hello. Good evening, Dashrath Hansa. So we are understanding the mechanism of enzyme action. The enzymatic reaction takes place by binding of the substrate with the active site of the enzyme molecule by several weak bonds. So this is how the enzymatic reaction takes place. It binds with the active site of the enzyme molecule by several weak bonds. So there is an enzyme. Let's see that if it is an enzyme, and this one is the active site. So over here the substrate will come. So this is the active site where the substrate will come. So it will lead to the formation of product, and enzyme will be replaced and product will be formed. So this is an active site. This area is known as the active site. Formation of enzyme substrate complex is the first step in the enzyme catalyzed reaction. Then enzyme substrate is subsequently converted to product and free enzyme. So after the reaction, the enzyme, this is the enzyme, this is one is the substrate. Just a minute. This one is the enzyme, this one is the substrate, and this one is the enzyme, and this one is the product. So product has been formed after the formation of enzyme substrate complex, and enzyme, free enzyme and product are being released after the enzyme substrate complex has been formed. So there are several mechanisms to explain this association, and lock and key or template model is an important hypothesis for understanding that how enzymes and how substrate get fits into the enzyme active site, and it is formed. So as you can see in this figure, so this is an enzyme and it is having an active site, and this is a substrate. Substrate has groups which are exactly same as the group that is present in the active site, and the substrate will get fixed just like a particular key has a particular lock, and substrate has a particular affinity towards the active site, so it gets fixed in the active site and it leads to the formation of the product. Then there is the induced fit model. So in the induced fit model, what happens? So there is a different this is another hypothesis which is known as the induced fit model. So substrate enter the active site, enzyme changes shape, so its active site embraces the substrate. Like whenever the substrate will come, the enzyme will change itself so as to accommodate the substrate, and the substrates are held in the active site by weak interactions such as hydrogen bond and ionic bond, and then the active site can lower the activation energy and speed up the reaction by acting as a template for substrate orientation, stressing the uh, substrate and stabilizing the transition state, providing a favorable microenvironment participating directly in the catalytic reaction, and substrate are being converted into the product. So as you can see that now the products have been formed and the free enzyme has been released. Now active site is available for two new substrate molecules in this case. So this is an induced fit model. According to induced fit, whenever the substrate will come, enzyme will change its shape so that its active site can embrace the substrate. So this is an induced fit model. Are you able to understand this induced fit model? If you are able to understand, please uh, type yes if you are able to understand the induced fit model. इंड्यूस फिट मॉडल में जैसे ही सबस्ट्रेट आएगा तो एंजाइम अपने आप इसका शेप अपने एक्टिव साइट का शेप चेंज करेगा, जिससे कि वो सबस्ट्रेट की जैसा एक्टिव साइट बना लेगा, और उसके बाद एक्टिव साइट में बहुत ही अपने सबस्ट्रेट के साथ वीक हाइड्रोजन बॉन्ड्स और आयनिक बॉन्ड के साथ उसको अपनी जगह पे हेल्ड अप रखेगा, और एक्टिव साइट लोअर करेगा activation energy and speed up करेगा rate of reaction by acting as a template for substrate orientation and stressing the substrate and stabilizing the transition state, providing a favorable microenvironment participating directly in the catalytic reaction. तो उसके बाद प्रोडक्ट बन जाएगा और एंजाइम सेपरेट होके बाहर निकल जाएगा. तो एंजाइम का भी एक्टिव साइट अवेलेबल होगा इनटू टू न्यू substrate molecule. This is the diagram where you can see that without it is showing energy on the y-axis and progress of the reaction on the x-axis. So here are the reactants. Here are the reactants. For example, H2O2 is the reactant. So this is reaction number 1, that is without the enzyme, and this is the reaction number 2 with the enzyme. And over here the products have been formed in both the cases, which is oxygen and water molecule. So without the enzyme the activation energy is very high. As you can see, the activation energy is very high. Without the use of enzyme, the activation energy is very high. बिना एंजाइम के एक्टिवेशन एनर्जी हाई है compared to the situation where enzymes are being used. जहाँ पे एंजाइम का यूसेज है वहाँ पे एक्टिवेशन एनर्जी कंपैरेटिवली काफ़ी कम है compared to उस रिएक्शन के जहाँ पे एंजाइम नहीं यूज़ हो रहा है, जहाँ पे एक्टिवेशन एनर्जी काफ़ी हायर है. And this is the overall energy that is released during the reaction. This is the overall energy that is released during the reaction. So as you can see that enzymes, they lower the activation energy of the reaction. So there is a nomenclature and enzyme classification. So IUBMB has recommended system of nomenclature for enzymes, and according to them each enzyme is assigned two names. One is the trivial name, that is the common name or the recommended name. Other one is the scientific name or the systematic name, that is the official name, sorry. So there are two names, दो तरह के नेम होते हैं, एक ट्रिवियल नेम हो गया, एक सिस्टमिक नेम हो गया. ट्रिवियल नेम जो होगा वो कि कॉमन नेम होगा और सिस्टमिक नेम होगा जो कि ऑफिशियल नेम होगा. तो सिस्टमिक नेम के बारे में कुछ चीज़ें हम लोग स्टडी करते हैं. यहाँ पे ईच एंजाइम इज कैरेक्टराइज्ड बाय अ कोड नंबर called enzyme code number or EC number and contains 4-figure digits separated by a dot. So first digit represents the class, second digit stand for the subclass, third digit stand for the subclass or the subgroup, fourth digit give the serial number of a particular enzyme in the list. So so that how you can see over here, this is one of the examples that how the EC number of the enzyme is given. This first one represents the class, second digit represents the subclass, third one represents the subclass or the subgroup, and fourth digit represents the serial number of a particular enzyme in the list. For example, in case of hexokinase, the number is EC 2.7.1.1 for hexokinase. So it means 2 is the class, 7 is the subclass, 1 is the subgroup, and 1 is the serial number of a particular enzyme in the list. This one is the systemic name. According to IUBMB system of enzyme nomenclature, enzymes are grouped into 6 major classes. There are enzymes that can be classified into oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. So EC1 are the oxidoreductase, EC2 are transferase, EC3 are hydrolase, EC4 are lyase, EC5 are isomerase, EC6 are ligase. This is the systemic name. तो गाइज आर यू अंडरस्टैंडिंग? आप लोग को समझ में आ रहा है जो भी बताया जा रहा है? अगर आप लोग को समझ में आ रहा है तो यस टाइप करिए कमेंट बॉक्स में. Now next we move forward toward the factors affecting reaction velocity. तो जो रिएक्शन वेलोसिटी है एंजाइम और का जो रिएक्शन वेलोसिटी होता है दैट इज डिपेंडेंट अपॉन देयर आर सम फैक्टर्स दैट इफेक्ट द रिएक्शन वेलोसिटी such as temperature, such as the pH, that is the hydrogen ion concentration, then substrate concentration, then enzyme concentration, then products of the reaction, presence of activators and inhibitors, then there are allosteric effects, and then there is time. So we will see one by one how these factors are affecting the reaction velocity. So this is an effect of temperature. So on the y-axis you can see the reaction velocity denoted by V naught. This one is the reaction velocity. And on the x-axis you can see the temperature. So as temperature increases, initially the rate of the reaction also increases, but at a particular temperature, that is the optimum temperature, rate of the reaction is maximum. The reaction rate is the maximum, and after a certain increase in temperature, the rate of the reaction falls. So there is an optimum temperature at which the enzyme works the best, and after that it becomes difficult for the enzyme to act, and it gets denatured, because as you know that enzymes are proteins and proteins get denatured on increasing the temperature. So there is a particular optimum temperature where this enzyme works. So this one is the T optimum. Next we move to the effect of pH. As you can see on the y-axis, again there is a mention of reaction velocity. On the y-axis there is a mention of reaction velocity, and on the x-axis there is a mention of pH. So over here you are seeing two different enzymes. One is pepsin, other one is trypsin, and this is the pH scale. So if you can see that pepsin requires an acidic pH range. It requires an acidic so pepsin requires an acidic pH to work at its optimum function, and trypsin on the other hand requires an alkaline pH. Trypsin on the other hand requires an alkaline pH. Pepsin requires a pH of somewhere around 1.8 to act at a particular reaction, and trypsin on the other hand requires a pH of around 8. So you can see that there is a difference between acidic and alkaline acting of the different enzymes. So there are enzymes which work best in the acidic condition, such as pepsin, and then there are enzymes which work best in the alkaline condition, such as trypsin. Now next comes the rate of the reaction, or velocity is directly proportional to the enzyme concentration when sufficient substrate is present. Whenever substrate is sufficient, then the rate of the reaction or velocity is directly proportional to the enzyme concentration. An accumulation of the product in a reaction causes inhibition of the enzyme activity. So if there is a reaction in which sufficient amount of substrate is present, then you will get a straight line uh, uh, where slope will be value of slope will be equal to 1, or 45-degree line, because over here the rate of the reaction is directly proportional to the enzyme concentration, because the substrate is sufficient and it's present in the sufficient amount. But if the product that is formed in the reaction causes inhibition, because sometimes what happens, that product gets accumulated in the reaction and it leads to the inhibition of the enzyme activity. कभी कभी क्या होता है कि प्रोडक्ट एक्यूमुलेट हो जाता है रिएक्शन के बाद, जिसकी वजह से इनहिबिशन हो जाती है एंजाइम एक्टिविटी का. Now we will see effect of substrate concentration. On the y-axis you can see the reaction rate. On the x-axis you can see the substrate concentration. So there is a maximum substrate maximum rate at which the substrate concentration there is a particular substrate concentration where rate of the reaction is maximum, and it's rate limiting in nature. And when the substrate concentration falls, the rate of the reaction also falls. But there comes a time when the substrate concentration is optimum and the rate of the reaction is maximum. And after increasing the substrate, after the optimum substrate concentration, there is no effect on the reaction velocity rate. Good evening, Pooja. Good evening. Now we will be studying something about enzyme kinetics. हम लोग enzyme kinetics के बारे में अब स्टडी करने जा रहे हैं. आप लोग के अगर कोई डाउट्स हैं तो आप लोग उसे कमेंट बॉक्स में टाइप करिए. Yes, Pooja, Dashrath, Marina, Shatruhan. If you have any doubt, you can type in the comment box. Shruti Jain, good evening. Now we will be studying something about the enzyme kinetics. I hope you are able to understand in a better manner. So the study of reaction rate and how they change in response to response in experimental parameters is known as kinetics. So the study of reaction rate and how they changes in response to change in experimental parameters is known as kinetics, and the amount of substrate present in is one of the key factors affecting the rate of the reaction catalyzed by an enzyme in vitro. So over here in this graph, on the x-axis we can see the substrate concentration, which is denoted by capital S, and on the y-axis we can see the initial reaction rate, which is given in micromolar per minute. So and this is the Vmax, where the maximum reaction rate is observed, and this is the Km value. Km value is that reaction rate where half of maximum Km is that substrate concentration where half of Vmax is obtained, and after increasing the substrate concentration there is no change in the Vmax value. So this graph denotes about that how the substrate concentration influences the initial reaction rate. Amit Atri, thank you Amit Atri. ये सब कुछ आप ही लोगों के लिए किया जा रहा है जिससे कि आप लोग इसको अच्छे से समझ पाएँ. आप लोग इस क्लास को बहुत सारे लोगों से शेयर करिए, जो भी नीडी है, जो भी पढ़ना चाह रहा, तैयारी कर रहा है इस एग्ज़ाम के लिए, आप उन लोग से शेयर करिए. तो ये यहाँ पर हम लोग ने देखा कि सबस्ट्रेट कंसंट्रेशन का क्या फ़र्क पड़ता है इनिशियल वेलोसिटी पर, एंड हम लोग ने वहाँ पे एक Km वैल्यू डिटरमाइन करी. Km वैल्यू वो सबसेट कंसंट्रेशन होता है जहाँ पे रेट ऑफ रिएक्शन हाफ होता है उसके Vmax के. Vmax होती जो मैक्सिमम वेलोसिटी ऑफ द रिएक्शन होता है, बट Km वो सबसेट कंसंट्रेशन होता है जहाँ पे हाफ रिएक्शन वेलोसिटी Vmax मिलती है. तो हम लोग आगे बढ़ते हैं. तो उसके बाद हम लोग स्टडी करेंगे माइकलिस मेंटेन काइनेटिक्स की. सो इस मॉडल में जो खास बात है वो है कि वो इट इन्वॉल्व्स वन सबस्ट्रेट मॉलिक्यूल. तो दिस इज enzyme is denoted by E and substrate is denoted by S, and there is a formation of enzyme substrate complex by ES, and then there is a formation of enzyme plus product. So these are the rate constant K1, K2 and K minus 1, where S is the substrate, E is the enzyme, and K1, K minus 1, K2 are the rate constant. So the mathematical equation that defines quantitative relationship between the rate of a enzyme reaction and the substrate concentration is known as Michaelis-Menten equation. तो एक Michaelis-Menten equation डिराइव होती है जिसमें V नॉट इज इक्वल टू वी मैक्स एस नॉट अपॉन के एम प्लस एस नॉट अपॉन के एम प्लस एस होता है. तो ये जो इक्वेशन है इसको हम लोग ऐसे भी डिपिक्ट कर सकते हैं. V नॉट इज इक्वल्स टू वी मैक्स इंटू सबस्ट्रेट कंसंट्रेशन अपॉन के एम प्लस सबस्ट्रेट कंसंट्रेशन. अगर हम ये इनिशियल रिएक्शन रेट है, ये मैक्सिमम रिएक्शन रेट है, ये Km है, Km that is the separate concentration where half of maximum reaction half of Vmax is obtained. तो अगर हम इस रिएक्शन को सिंप्लीफाई करें तो हम लोग यहाँ पे लिख सकते हैं 1/V0 इज इक्वल्स टू Km by Vmax just a minute. हम लोग अभी आगे देखेंगे एंड ये Km मिलता है जिसकी वैल्यू हम लोग कैलकुलेट करते हैं बाय K-1 प्लस K2 अपॉन K1. So this is an effective Michaelis-Menten rate constant. अब हम लोग जो ये इक्वेशन थी V naught is equals to V max into S upon Km plus S. एक लाइन वीवर बर्ग प्लॉट होता है. लाइन वीवर बर्ग प्लॉट एक डबल रेसिप्रोकल प्लॉट होता है एंड वो एक लीनियर रिप्रेजेंटेशन होता है जो कि मोर एक्यूरेट होता है एंड कन्वीनियंट होता है फॉर डिटरमाइनिंग वी मैक्स एंड के एम. तो अगर हम लोग को वी मैक्स और के एम डिटरमाइन करना है तो हम लोग लाइन वीवर बर्ग प्लॉट के लिए जाते हैं और ये जो इक्वेशन है ये ऑब्टेन होता है बाय टेकिंग द रेसिप्रोकल ऑफ बोथ द साइड्स ऑफ द माइकलिस-मेंटन कांस्टेंट. तो अगर हम लोग को रेसिप्रोकल लेना है तो हम लोग कैसे प्रोसीड करेंगे, तो ये हो जाएगा 1/V0 इक्वल्स टू Km अपॉन Vmax इंटू 1/S0 1/S प्लस 1/S 1/Vmax. So this is the reaction. और ये जो रिएक्शन है इसको हम लोग एक स्ट्रेट लाइन की जो इक्वेशन होती है y इज इक्वल्स टू mx प्लस c से डिपिक्ट कर सकते हैं. मैं इस इक्वेशन को फिर से इस तरफ समझाता हूँ एक बार, यहाँ पे कुछ लोग को देखने में परेशानी होगी. जस्ट अ मिनट. सो इस इक्वेशन को हम लोग डिपेंड कर सकते हैं y इज इक्वल्स टू एम एक्स प्लस सी से, और हम लोग एक स्ट्रेट लाइन पे प्लॉट कर सकते हैं जहाँ पे स्लोप होगा, जो स्लोप होगा लाइन का वो होगा के एम अपॉन वी मैक्स। एक्स एक्सेस पे आ जाएगा 1/s0, 1/s, और वाई एक्सेस पे आ जाएगा 1/v0। तो हम लोग नेक्स्ट पेज पे चलते हैं। तो जैसा कि आपने देखा, जो स्लोप होगा लाइन का वो के एम अपॉन वी मैक्स से डिपिक्ट किया जाएगा। जो स्लोप है इक्वेशन की वो डिपिक्ट की जा रही है। ये एक डबल रेसिप्रोकल प्लॉट है लाइन व्यूअर ब्लॉक प्लॉट का। ये कैसे आया है हमने आप लोग को बताया। और यहाँ पे जो स्लोप होगा वो के एम अपॉन वी मैक्स से डिपिक्ट किया जाएगा। एक्स एक्सिस पे सबस्ट्रेट का कंसंट्रेशन मिली मोलर में आ जाएगा, और रिएक्शन रेट माइक्रोमोलर पर मिनट में 1/v0 से दिखाया जाएगा। ये इंटरसेप्ट है, इसकी वैल्यू होगी 1/v मैक्स। ये वाई इंटरसेप्ट है, इसकी वैल्यू होगी 1/v मैक्स। ये एक्स इंटरसेप्ट है जिसकी वैल्यू होगी -1/km। तो इससे हम लोग km और v मैक्स की वैल्यू डिटरमाइन कर सकते हैं। इससे हम लोग स्लोप ऑफ द रिएक्शन निकाल सकते हैं। नेक्स्ट हम लोग आते हैं एंजाइम इनहिबिशन पे। कभी कभी क्या होता है कि कुछ सब्सटेंस दे डिमिनिश द वेलोसिटी ऑफ एन एंजाइम रिएक्शन। These include drugs, antibiotic, poisons, and antimetabolites, and they are useful in understanding the sequence of the enzyme catalyzed reaction, metabolic regulation, studying the mechanism of the cell toxicity produced by toxicant, and it forms the basis of drug designing. तो एंजाइम इनहिबिशन में कभी कभी क्या होता है कि कोई कोई सब्सटेंसेस ऐसे होते हैं जिससे कि जो रिएक्शन की जो वेलोसिटी होती है वो डिमिनिश हो जाती है। रिएक्शन की वेलोसिटी है वो डिमिनिश हो जाती है बिकॉज वो एंटी मेटाबोलाइट्स होते हैं, एंड दे आर वेरी हेल्पफुल इन अंडरस्टैंडिंग द सीक्वेंस ऑफ द एंजाइम कैटालाइज रिएक्शन, metabolic regulation, and studying the mechanism of cell toxicity produced by toxicants. They form the basis of drug designing. तो enzyme inhibition एक important concept है, और enzyme inhibition बेसिकली दो तरह से होता है। एक होते हैं रिवर्सिबल इनहिबिटर्स, एक होते हैं रिवर्सिबल इनहिबिटर्स, एक होते हैं इरिवर्सिबल इनहिबिटर्स। रिवर्सिबल इनहिबिटर्स फर्दर क्लासीफाई होते हैं 3 टाइप्स में: कॉम्पिटिटिव, नॉन कॉम्पिटिटिव और अनकॉम्पिटिटिव। तो गाइज मैं आप लोग को एक बार फिर से बताना चाहूँगा कि आप लोग अनअकैडमी, आप लोग को क्लास कैसी लग रही है। आप लोग टाइप करके कमेंट बॉक्स में बताएं। और मैं आप लोगों को फिर से बताना चाहूँगा कि आप सभी लोगों को मैं वेलकम करता हूँ अनअकैडमी के प्लेटफॉर्म पर। आप लोग अनअकैडमी के प्लेटफॉर्म पर बहुत सारे एजुकेटर्स के, टॉप एजुकेटर्स के कोर्सेज को पा सकते हैं, जिससे कि आप अपना जो गोल है वो अचीव कर पाएंगे CSIR नेट क्रैक करने का। तो उसके लिए आपको क्या करना है, उसके लिए आपको गूगल प्ले स्टोर पे जाना है। गूगल प्ले स्टोर पे जाने के बाद अनअकैडमी का लर्निंग ऐप को आपको इंस्टॉल करना है। आपको अपना गोल सेलेक्ट करना है, और गोल सेलेक्ट करने के बाद आपको CSIR UGC नेट की कैटेगरी आ जाएगी, जहाँ पे आपको वेरियस टॉप एजुकेटर्स के कोर्सेज मिलेंगे। अगर आपको एक्सक्लूसिव गाइडेंस चाहिए, यू नीड टू सब्सक्राइब फॉर द CSIR UGC नेट कैटेगरी। आपको सब्सक्रिप्शन लेना पड़ेगा। सो वंस यू टेक द सब्सक्रिप्शन फॉर CSIR UGC नेट, the subscription is available for 1 month, 3 months, 6 months, 12 months, and 24 months. So I would recommend that you take the subscription for 12 months to 24 months because classes year long चलेंगी and आप लोग को इसका भरपूर फ़ायदा मिलेगा. आप लोग अच्छे से एजुकेटर के साथ इंटरेक्ट कर पाएंगे, उनको समझ, उनके मेथड्स को समझ पाएंगे और अपने एप्लीकेशन को नॉलेज के एप्लीकेशन को अप्लाई कर पाएंगे इन सॉल्विंग वेरियस मॉक टेस्ट एंड क्विजेज. So you can always use my referral code HY10, and this is for an academy subscription, CSIR UGC NET subscription. So you need to continue, and आप लोग जब continue करेंगे, so you need to confirm your payment details, and you can make the payment either by card, debit card or credit card, or by EMI or by net banking. And and after applying my code HY10, you will get the entire classes for 10,395 instead of 11,550. So let's go back and study where we were actually. So we were understanding the case of reversible inhibitors where they are classified into three categories such as competitive, non-competitive and uncompetitive. So in case of competitive inhibition, what happens? Like there is an enzyme and then there is a substrate. So a competitive inhibitor will come and it will bind with the enzyme, and competitor inhibitor जो होगा वो substrate को mimic कर रहा होगा. कॉम्पिटिटिव इनिबिटर जो होगा वो सबस्ट्रेट को मिमिक करेगा, वो सबस्ट्रेट जैसा ही होगा और वो एंजाइम में जाकर एंजाइम के साथ इनिबिशन करके एंजाइम इनिबिटर कॉम्प्लेक्स बना लेगा. एंजाइम इनिबिटर कॉम्प्लेक्स बना लेगा एंड फर्दर रिएक्शन हो नहीं पाएगा. नॉर्मल रिएक्शन में क्या होता है कि एंजाइम सब्सट्रेट कॉम्प्लेक्स बनाते और फाइनली में प्रोडक्ट और एंजाइम मिल जाते. बट अगर इनिबिटर आ जाएगा तो इनिबिटर के आई उसका रेट कांस्टेंट कोफिशिएंट होगा और वो एंजाइम के साथ एसोसिएशन बना लेगा, अपना ई आई एसोसिएशन बना लेगा और सबस्ट्रेट अलग हो जाएगा और कोई फर्दर रिएक्शन नहीं हो पाएगा. ये केस है एक कॉम्पिटिटिव इनिबिशन का, जहाँ पे कॉम्पिटिटिव का मतलब है जहाँ पे कॉम्पिटिशन हो रहा है बिटवीन द सबस्ट्रेट एंड द कॉम्पिटिटिव इनिबिटर. दिस वन इज द केस ऑफ नॉन कॉम्पिटिटिव इनिबिशन. यहाँ पे जो कॉम्पिटिटिव इनिबिशन के केस में मैं आपको बताना चाह रहा था, यहाँ पे जो कॉम्पिटिटिव इनिबिशन जो हो रहा था उसमें जो कॉम्पिटिटिव इनिबिटर था उसकी एक्टिव साइट के साथ सेम एफिनिटी थी जो कि सबस्ट्रेट की होती है. बट इन केस ऑफ नॉन कॉम्पिटिटिव इनिबिशन जो एफिनिटी जो नॉन कॉम्पिटिटिव इनिबिटर होगा वो एक्टिव साइट को छोड़ के किसी अदर साइट पे एक्ट करता है. तो जैसे either it can bind to the enzyme or it can bind to the enzyme substrate complex and it can halt the reaction. So no reaction will take place. So यहाँ पर in this case of non-competitive inhibition, the enzyme can bind to the inhibitor forming the enzyme inhibitor complex, or it can bind to the enzyme substrate forming the enzyme substrate inhibitor complex, and no reaction will take place. And then there is the uncompetitive inhibition where it binds only to the enzyme substrate complex and it does not have the capacity to bind to the free enzyme, and it is also not overcome by increasing the substrate concentration. So both Km and Vmax value are reduced in this case. This is a case of uncompetitive inhibition. Samajit Patra, good evening. माइकलिस मेंटेन इक्वेशन मैंने आप लोग को बताई थी। माइकलिस मेंटेन इक्वेशन डिपिक्ट होती है V naught is equals to V max S naught upon Km plus S naught से. This is the Michaelis Menten equation. Samjit, this is the Michaelis Menten equation. This is the Michaelis Menten equation. So it defines the quantitative relationship between the rate of an enzyme reaction and the substrate concentration. This is known as the Michaelis Menten equation, where V naught is equals to V max into S upon Km plus S. This is the Michaelis Menten equation. Okay, Samjit, any other doubt? So this is the case of uncompetitive inhibition, where the enzyme and the enzyme substrate complex is formed, and the inhibitor comes and it blocks the any other site apart from the active site of the substrate, and it forms the enzyme substrate inhibitor complex. So these are the double reciprocal plots. In case of competitive, non-competitive. So in case of competitive inhibition, what happens? So this is the normal equation. So when the inhibitor is not there, Vmax is obtained. So when the inhibitor is not there, Vmax is obtained. But when the inhibitor comes, so it lowers the Vmax value, and also a new Km value. Km is increased and Vmax remain unchanged. Vmax remain unchanged in this case, and Km is increased. As you can see over here, the value of Km is increased in case of competitive. So this is the inhibitor. As you can see that it intersects at y-axis and it forms a Y in case of competitive inhibition. So in case of competitive inhibition, Km is increased and Vmax remain unchanged. In case of non-competitive inhibition, what happens? The Vmax is decreased and the Km remains unchanged. So in this case, as you can see over here, the inhibitor is there and the value of Km is unchanged, because both the values of inhibitor and are intersecting at the same point, so Km remains unchanged and Vmax is decreased. Next is the value of uncompetitive inhibition, where both Vm and Kmax are decreased. So in case of uncompetitive inhibition, both Vm and Kmax are decreased. So we get two parallel lines instead of intersecting lines. So these are the enzyme inhibition plots. So guys, thank you. आप लोग के अगर कोई क्वेरीज हैं, कोई डाउट्स हैं, तो यू कैन पोस्ट. Krishnaveri, Vijay Kumar, morning and evening question papers were different. This we have to see. I think it will be different. अभी हम लोग को ये देखना होगा. Guys, please like and share and subscribe to this channel. and please share with your friends as much as possible, because it's time for a learning revolution here on Unacademy. And you can use my referral code HY10, where you will be getting 10% off on your next Unacademy subscription. So guys, please do like and share with your friends. अगर आप लोग की कोई क्वेरीज हैं, कोई डाउट्स हैं, तो उसको आप लोग पोस्ट करिए कमेंट बॉक्स में. नाउ द सेशन इज ओपन फॉर क्वेश्चंस एंड आई विल बी मोर देन हैप्पी टू आंसर देम. आप लोग के जो भी सवाल हैं, आप लोग इसको कमेंट बॉक्स में टाइप करिए. आई विल ट्राई टू आंसर देम. यस गाइस, आपकी तरफ से कोई क्वेश्चन नहीं आ रहे हैं. अगर कोई क्वेश्चन है तो उसे कमेंट बॉक्स में टाइप करिए. इफ़ यू हैव एनी काइंड ऑफ डाउट, यू कैन पुट इन द कमेंट बॉक्स. प्लीज़ आस्क योर डाउट्स. So I don't think that there is any doubt. So let's stop the session.